It's really my pleasure to be here in my own country, a different place. And we've been, as Jerry said, to mountains, now to the sea. Maybe we could be in my own town next to the Danube River someday. I'll talk to you about aquaphotonics, and I'll start with introduction of two things. We use spectroscopy to analyze water, and I'll say a few words about near-infrared spectroscopy, and then I'll tell you what aquaphotonics means. And then I'll go to two papers that we published by chance, I don't know by what, on the same date. We got accepted on July 2nd. There was a paper in Nature's online scientific reports and in PLOS ONE. So, as Alberto said this morning, we have to be strategically right and get some papers in well-read journals, and then start by this telling a bit more about what we know for water. I'll start with this slide because aquaphotonics is all about aqua is water, and this is the first character, Japanese character, and sounds Mizu. And the second character is water, it's light, which is hikari in Japanese. So, near-infrared spectroscopy. Now, in medical science, it is used not traditionally as any other spectroscopy quantitatively to measure components. It is used for monitoring. And I think we have to stress, it was mentioned just this morning, is very important as a dimension. So we can measure, but if we add time, we add lots of information. So these people use infrared light for pulse oximetry. This is quantitative approach, so they use only 2-3 wavelengths, 2-3 energy levels, because they penetrate very well in human tissue. And they use infrared light because it penetrates very well also into the tissue to follow the oxyhemoglobin ratio, which means the functioning of brain areas, which is you can do function near-infrared instead of functional MRI in order to follow which part of the brain are activated. This is possible only because near-infrared light penetrates. It is not fully absorbed by the water as infrared light is. It is partially absorbed, which gives us the possibility to analyze the light coming back from the water or aquasystem or biological system. So this light is invisible for us. It is next to the red light. It has less energy than the visible and UV. And it has a specific, in the electromagnetic spectrum, this is infrared light. But this is near-infrared, which is from 700 to 2500 nanometers. So very short, comparatively speaking, range. Specifically this near-infrared, we call it next to the visible. Some people call it far visible. The beauty is that it really penetrates about up to 10 centimeters into aqueous or water system or biological system. So this is the spectrophotometers, how they look. We can use fibers. We have used fiber probes and handheld instruments to analyze different biological systems in vivo monitored. Okay? So what we found, these are serious, I'm just jumping because I want to go to the content of my presentation, but this is just so many systems that we have analyzed with the spectroscopy, and we have found that we can use the spectral of the system over the time, the change of the spectral pattern of the system as a biomarker. So we could use this spectral pattern for diagnosis. And this is an interesting, this is inflammation of udder in cows. This is prion disease in mouse. And I will relate this to Luc Montanier's talk. We found prion disease, we were able to diagnose prion disease. We inoculate 40 mice for 2 years, 2 different experiments every year. And we consistently found out that we were able to diagnose the disease, firstly, in the abdomen, not in the brain, where you would expect to find prion disease. So it is a lot to understand in the future about what actually is causing the disease. And HIV again, HIV diagnosis, we can do this on plasma, on serum. So, and this is the spectral pattern. And the specific spectral pattern that relates to oxidative stress, as Luc mentioned. So, one interesting thing is that near-infrared is an overtone spectroscopy. Overtone means that vibrations, it is not the main vibration with the highest amplitude, which is in the infrared region. This is the second, third, and fourth amplitude, like a bending. This is the Hooke's law. The amplitude goes 100,000, 10,000 times less, which means that if we have a component, a molecule that is at very low concentration, we are not able to see and to measure this molecule by near-infrared vibrations of the molecule in the near-infrared range, because it's a very low concentration. But you will see, we can measure. We can make a perfect models only if we use absorbance of the water. And by all this analyzing all the systems, what we've found out that we were able to diagnose that diseases only by looking at the spectra of the water. Because what is changing is that water is playing as a molecular mirror when illuminated with near-infrared. You know that water is a mirror for the visible light. Water is a mirror as well with near-infrared. So all the molecules are changing the water structure. They're changing the strength of the hydrogen bond and the covalent bond. The strength and the angle in the molecule is changing because of the surrounding other molecules. Now, this is the spectra of the water. I'm sure you don't see anything, but if you analyze the spectra, which is the number 2 requirement, is the multivariate analysis of all the spectral data that we have. We have to have a lot of spectral data for many samples to make the model. But if we analyze this, we could see many absorbance bands underneath. We could see that really water is not just one single molecule with one covalent bond. We could see that water is a system, is a molecular system. And the strength of the covalent bond is changing, and it's following the environment, as you want to call it, epigenetics or whatever. Now, probably you'll understand easily, this is the spectra of the sun, the intensity of the sunlight. Volodya says that we know lots of water found in the space. You see, this is the maximum of intensity of the sunlight coming measured above the atmosphere. He is above the atmosphere. He is on the sea level. This is the visible light. This is from 400 to 700 nanometer. Visible. So the highest intensity, the lowest is here. The half of the spectra of light, of sunlight, is near-infrared light. Half of the spectra. So, and you see, we don't get all the light, the sunlight, because it's absorbed by the atmosphere. But you could see that it is also absorbed by the space from the sun to the earth. So there is water. This is the spectrum of water. But this absorbance spectra. How much light water absorbs? It absorbs a lot in UV. It absorbs nothing, nearly invisible. So that's why it's so bright outside. And infrared absorbs a lot. So to use infrared, we need a very thin samples. But infrared has a specific spectral pattern. And also we can monitor, we can use native samples. We don't need preparations, we just can do this non-invasively. Now, so what we have to pay attention is about this hydrogen bonding and the covalent bond. That strength, as I said, and angle depends on the environment. Now, water's molecular mirror. And today we started with reductionism and systematic approach and analysis and synthesis. I think that these are the two sides of the coin, so as yin yang, and so we have to have them both, I think. But aquaphotonics gives the other side. We don't go molecule by molecule, we just look at the system and we have a holistic approach to the system. We could, by looking at all water molecules, the water molecular system, and that, as I said, other molecules are mirrored by water, so we could have the idea and knowledge and numbers, so we could measure what's happening with the system as it is. So we talk about Albert St. George, and also in 16th century, it is said that water is a matrix of the world, and we have to pay attention to the two matrices, water and electromagnetic field. And this is what we propose with aquaphotonics. We introduce perturbations to the system. We activate, because water is such a sensitive conglomeration of molecules. It gets the influence of the temperature strongly, humidity, pressure, everything. So if we talk about epigenetics, it goes directly to the water, and water influences DNA and etc. So probably, and it is vice versa. It is a play. But in order to use this spectroscopy, what we have to know to have the database of water absorbance bands. And I call them water matrix coordinates. At those coordinates means windows. At those coordinates, we can measure water, and we need perturbations to activate those bands in order to change entropy, to get more information for the system. So, water matrix coordinates, WOMACs, and we found we've concentrated under the first overtone. We know much more about the second and the third already, but under the first overtone we found 12 coordinates, 12 bands that we could use as a point to transfer information. And we could identify those bands with different water structures. Now, I know we get nervous when we say what is structured water, and do we understand what is structured water at all? No, we don't. And I cannot say that I understand really. But we could see that we have this conformations like we have four molecules of water. On the top of them, if we have this OH, then this covalent bond has a different absorbance band, a specific band. The same for 2, for 1. Also, we have, if here we have oxygen, 2 oxygen, we have different band. So these are coordinates of specific water structures. And what is structure, what is a different discussion, I think. So aquagram is a graph that we use to express what is the water profile, what is the water molecular profile seen by the absorbance of light of water-specific bands. Now, this is the spectral vapor, and this is the spectral water. You see that they overlap each other. So in the water, we have vapor and ice at the same time, but in different ratio. And the ice spectra is somewhere here. Now, maybe if you remember now, the right side of this aquagram, this is the high energy bands, absorbent bands of the water. This is the less hydrogen bonded water. If we talk about for example, when you increase the temperature of water, less hydrogen bonded water increases, and hydrogen bonded water decreases. Ice decreases the vapor part. The vapor part is the left high energy part, this one. This absorbance increases here, and in ice, this absorbance increases, this part. Okay. Now, when we increase the temperature, as I said, this is the spectrum moves, has a blue shift. When we decrease the temperature, it has a red shift. So this is the right side of high temperature water, and this is the low temperature water, aquagram. Now, I'll talk about UV-induced changes of the DNA. What we did, this is a paper that I told you is the first one, published on July 2nd. But before that, I will show you, before this study, what we did, we used 2 different circular double-stranded DNA and linear double-stranded DNA. We took spectra, the same concentration. Okay, you can't see anything. Sorry about that. So you'll believe me. I'll tell you. Okay. The spectra were different for different DNA, but this is what we did. We subtracted molecule and of this DNA, and we diluted it. And I just wanted to show when we do so what happens. This is so-called free water molecules, 1410, 1416 area. This is the ice clusters with 4 hydrogen bonded hydrogen bonded water. So what you can see is when we dilute or when we subtract molecule with DNA has more free water. This is very interesting. This is water solvation shell, specific band, specific profile. So what DNA is doing is cutting hydrogen bond, decreasing the hydrogen bonded part of the water, and increasing the less hydrogen bonded part. Now, I want to tell you, this is a new thing that I was thinking for the last few years gradually, but I needed more and more consistent results. Now, when I think of water, I imagine as 2 parts. This is, I called the motor part, working part. And this one is the battery part, the energy storing part. And there is a very important part, which is in between. Here you don't see it. Which is the buffer. Those are the water molecules. Probably this is highly related to what Emilio and people talking about coherent zones. So this is so important. The molecules that are moving from one zone to the other, from one coherent zone to the other, they are those that are actually stabilizing the water. Water is self-organizing system, and this self-organization is coming from these water structures that are keeping the balance between the two, the motor and the battery. Now, if we subtract the second dilution, so we see, you could see already C2, C3, C4, these are the water matrix coordinates, I call them, these are in nanometers, and each of those coordinates is related to a certain function. We don't know them all, of course, but I've seen them in different implants, in cells, adding different molecules as perturbers. And increasing one or the other, you see, like D5, you have 10 to minus 5 dilution, and you could get both. You could have highly organized water and also less hydrogen bonded water, mostly on the same level. But you lose this medium, the low-level members of the medium, the transition part. And all this is related to function, to different functionality. Okay, now the paper is what we showed. We showed that we look at this. So we work with micromolar concentration of DNA. There is no way that near-infrared light can measure the molecule itself. So we were able to see this high correlation to make a model only because we were looking only at the first overtone of the water. And you could see that when we increase or change the concentration of DNA, we have positive, as I showed you in other DNA studies, we increase the less hydrogen bonded part of the water. Now, if we introduce UV light to the sample with DNA, you see that this is the control, and the red is UV light emitted sample. And these are the water absorbing bands that are working. So because of changes in this area, we get this separation. So we could identify what's happening. Now, this is a model, a beautiful correlation between the UV dose from 0 to 20 kilojoule per square meter. And look at this, what happens? Reverse. UV light is ordering water molecules. UV light is making water structure. Is making more hydrogen bond in water and less hydrogen, non-hydrogen bond in water. Now, this is the concentration of the dimer produced by DNA as a result of the UV radiation. Now, the same effect, but we were able to measure with that accuracy with light interacting with water. Much better than HPLC. You can go to the paper, so we compared. We measured by HPLC, we measured by infrared, and you could see that infrared light can see the changes in the water caused by the modification of DNA as a result of UV. Now, another paper was bacterial functionality. So I liked what Luke said this morning for probiotics and non-probiotic. We looked at non-probiotic bacteria is the blue, moderate is green, and probiotic is red. Now we wanted to see, because you have a microorganism, a new microorganism, you want to analyze to see if it is a probiotic, if it has a strength of survival, etc. It takes months, even year. Now, with this profile, we can analyze, we need only 10 hours to say if this particular strain is probiotic or non-probiotic or moderate. This is from a practical side. From a theoretical side, what I would say, you could see that these are the loadings first and second, and the first and second loading, you could see the wavelengths that are activated. So you see, if you remember, this is water solvation shell. This is ion hydration band. This is S1, which is 2 water molecules with 1 hydrogen bond, very responsible for the stability of the water system, molecular system. And this is ice cluster or hydrogen bonded water. Now, this is the aquagram. These are the probiotics. If you remember, when you increase the temperature, we get less hydrogen bonded water, and we have absorbance on the right side. So what we have with probiotic bacteria is just working, is making water a working water. And a non-probiotic bacteria is not that bad. I mean, it doesn't go the whole side like this, but doesn't have these free water molecules, which are nearly very active, mostly activating, I would say. And these are the moderate, they have both. They have these free water molecules, and they have hydrogen bonded water structures that are potentially a kind of potential for these actions here, but they have different, specific spectral pattern. So they are in somewhere in between. And in the paper you can find assignment of all activated bands by this bacteria. So what we do, and I feel, at that time I feel very happy because there are many papers already in infrared spectroscopy, using infrared spectroscopy, and very low number of water molecules at different conformations. So they identify the exact absorbance bands of those structures, as I showed you before. When we calculate the overtone of these bands, and when we see these overtones exactly in the place where our experiments show high variations, that's the happiest moment in my life. So you see that experimental data proves theoretical calculations. So, and these are the structures, and these are the bands that we found, calculated as wave numbers. This is the calculated reference bands from references, and this is the exact structure found in this reference. So you could go to the reference, you could see what the structure means, and you could see where we can find it in the infrared. And now I have 5 minutes. I prepared this promptly while listening to Luke and Jerry, and so I just wanted to show, to link a little bit to what I was talking. I think Luke, we did an experiment, and we got very good results. We presented this, but we have not published it. But we have already this paper for DNA, and I think we're on the way. We can write already. So that was a strategic way to have something that people really understand. But this is the oxidated cells. The blue spectra are oxidated cells. So this is what they don't have is this transition state, and what between motor and the battery, and they have lots of, they will understand what exactly is there, but we could see what has been published already by Jerry's group, then light is changing the water. And this is my group. And okay, I'll finish with this. Thank you.